Hi all, this is CATCH25 Science Academy YouTube channel and you are watching Biology Snippets with Vrushti Mehta. In this Biology Snippet series, we will regularly upload short videos overviewing interesting topics from different chapters of biology. Today's topic is called Biomolecules, Part 1. But before we start with the video, I request you all to kindly subscribe to our channel and share our videos. Do not forget to like the video and press the bell icon for further notifications if you like our content. So, let us get started. Every cell and therefore tissue is made up of fundamental chemicals called biomolecules, which perform basic functions in a living organism. Now, if we wish to know what exactly is present in these living tissues, we'll have to analyze that particular tissue chemically. Let us see how this is done. First, take any living tissue, like a vegetable or a piece of liver. Take trichloroacetic acid liquid, grind them together in a mortar and pestle. This gives a thick slurry after some time. Now, like we filter tea at home, this thick slurry is also filtered through a cloth. The liquid part of the slurry passes through the cloth is called the filtrate, and the part which gets trapped on the cloth is called the retentate. The chemicals which do not dissolve in the acid and remain on the cloth are labeled as acid insoluble fraction. When grinding of tissue was being done, some chemicals from the tissue must have dissolved in the trichloroacetic acid. These acid-soluble chemicals which passed through the cloth with acid were hence called as acid-soluble fraction. When the chemical analysis was done, the acid-insoluble part was found to have carbohydrates, proteins, nucleic acid and lipids. These chemicals are known as biomacromolecules or simply macromolecules. On the other hand, acid-soluble part was found to have thousands of organic molecules called micromolecules, and few inorganic compounds like sulfates and phosphates were also found. So this is how biomolecules are extracted, isolated and purified. Such analysis can play an important role in identification of molecular formula and structure of compound. After hearing acid soluble and acid insoluble a million times, let us try to differentiate them now. Acid insoluble fraction contains carbohydrates, proteins, nucleic acid and lipids. Acid soluble fraction, also known as the extract, contains thousands of organic molecules and inorganic compounds. Carbohydrates, proteins and nucleic acid weigh generally more than or equal to 10,000 daltons and lipids weigh less than 800 daltons. The molecular weight of acid soluble fraction ranges between 18 to 800 daltons. Acid insoluble fraction represents macromolecules of cytoplasm and cell organelles of cells of the tissue used. Acid soluble fraction represents contents of cytoplasm of cell of the tissue used. All molecules in the acid insoluble fractions are polymers except lipids. In the last slide, we saw lipids were categorized under macromolecules, even though their molecular weight is low. Let us understand why is that so. We very well know that lipids are an important part of our cell membrane. When we grind a tissue during chemical analysis, cells which are in the tissue will break. Due to this, lipids present in the cell membrane of these cells will also be released. These lipids, after being released, are found as water-insoluble vesicles. Because they are water insoluble, they will not dissolve in the trichloroacetic acid, which we take during our experiment. Hence, you can make out why lipids do not get filtered with the acid. This was all about the analysis of tissue for organic biomolecules. What if we want to analyze inorganic chemicals? Let us see how that is done. Again, we start with a living tissue. Why? Answer is simple. We start with a living tissue because we are analyzing biomolecules, remember? So first step is to weigh tissue. Note this weight as wet weight. Now dry the tissue by evaporating the water content of the tissue. Weigh the tissue again. Note the weight as dry weight. Next step is we burn the tissue in the presence of oxygen. Let us recollect that this tissue which is being burnt has both organic and inorganic compounds in it. All the carbon-containing organic compounds will be lost in gaseous form, such as carbon dioxide and water vapor, while the inorganic compounds are obtained in form of ash. In a nutshell, we have two types of analysis, compound analysis and elemental analysis, as summarized in the table. So, that's it from today's biology snippet. If you found this content helpful in understanding the topic in a better way, subscribe to our channel for more videos. Thank you.